All right, hi everyone. This is the fourth video lecture on the topic cytoskeleton. In the last video, we talked about the actin filaments, and in this one, we're going to talk about the intermediate filaments. We're going to talk about the protein monomers that make them up, how they assemble into long filamentous intermediate filaments. In this case, we're not going to talk a whole lot about how they were regulated, but we will talk about one particular fundamental role that they play in cells. All right, so when you think of intermediate filaments, you can really think of many different intermediate filament proteins. There are several of them in cells, and they all share a common structure. That structure is depicted here. So it turns out that all of the different intermediate filaments all consist of a central rod domain, which is shown here. And so that central rod domain is usually just an elongated alpha helix domain of about 300 to 350 amino acids in length. And then on either side of that central rod domain is a more globular N-terminus that's sometimes called the head domain, and a more globular C-terminus, which is sometimes called the tail domain. So for all intermediate filament proteins, the amino acid sequence that makes up this central rod domain is very similar. And so we could say that this central rod domain is in common with all of the intermediate filaments. However, it turns out that the particular amino acid sequences and the structures that the head and tail domains adopt can be quite different between the different intermediate filament proteins. And that is really what dictates the specific functions for each individual intermediate filament protein. To start thinking about how these things assemble to form filamentous structures, we can say that they will assemble head to tail in coiled coils to form the filaments. So to try and illustrate what I mean by this, I'm going to use this image from your textbook. This is just showing you what we just looked at, just a smaller view, and we're going to show how these things come together to form long filaments. The first thing that happens is two individual intermediate filament monomers come together to form a dimer, and they do so by forming a coiled coil structure as is depicted here. So that central rod domain basically wraps around each other to form this coiled coil. Then two dimers associate in a side-by-side manner, though a little bit staggered, and in reverse orientation, as you can see, in terms of their head domains and their tail domains. They'll come together side by side into this tetramer. And then these tetramers start aligning head to tail, as is shown here, to form what is called a protofilament. These protofilaments then start aligning alongside to each other, and as they're aligning alongside of each other, they also sort of wrap around, forming this almost cable-like structure that represents the intermediate filament. So that's basically it in terms of how these things are assembled to form the long filaments within cells. Like microtubules and actin filaments, the assembly of intermediate filaments is reversible. And so when cells build these things up, they can also break them down back into their simpler subunits. They don't do so as readily as they do microtubules and actin filaments, but nevertheless they can. And I say that because it's going to be important to keep that in mind when we get into talking about the cell cycle. Okay, the last thing we want to do then is just talk about what role the intermediate filaments play in cells. And really these things are also much simpler compared to actin filaments and microtubules in the roles they play. Because really the main thing that they do is they provide cells with structure. So for example, the intermediate filaments, there are particular intermediate filaments that form what we call the nuclear lamina. And recall from Biology 121 that the nuclear lamina is those cytoskeletal proteins that underlies and supports the nuclear envelope as is depicted here. So this is supposed to, of course, be a close-up of the nucleus inside this cell. Here's the nuclear envelope that defines the outer boundary of that nucleus. And if you look underneath, there's this sort of intertwined structure that underlines the nuclear envelope. This is supposed to represent the nuclear lamina. And the nuclear lamina is composed of intermediate filaments that are built up just like we just went through. And they sort of underlie the entire nuclear envelope, which provides it with its structure as well as stability. So when we get into talking about the cell cycle, we're going to talk about how the modification or the breakdown and buildup of these intermediate filaments is key during the progression of the cell cycle. Okay, and that's it then. We've talked about actin filaments, intermediate filaments, and microtubules, And that closes off our discussion of the cytoskeleton.